And he is going to tell us about incredibly efficient protein ionization, which of course is of great interest for us because ionization efficiency is one of the main challenges for doing single cell proteomics by mass spectrometry. So I look forward to see how you accomplish that feat.
Thank you. My slides are on this computer. Okay, great. So hello everyone, my name is Nicholas Drachman. I'm a PhD student in the physics department at Brown University, working with Professor Derek Stein. I'm excited to be sharing some of our work on developing an ion source for single molecule protein sequencing. And much of what I'm going to present today was just published yesterday on bioRxiv, which you can find at the link here. Okay, so this talk is going to be a little bit different from most of what we've heard so far, because what we're working towards is not just single cell analysis, but single molecule analysis. So why do we care about being able to sequence proteins at a single molecule level? Well, these past 2 days, we've heard about some remarkable and impressive progress on sample preparation and data acquisition and analysis pipelines for single cell proteomics. But there are still some problems that conventional MS proteomics will never be able to solve. For example, there may be some biologically or clinically relevant biomarker proteins, or PTMs, that are present in as few as 10 copies per cell. And with no way to amplify these proteins, we need to be able to sequence them at a single molecule level. Now, there are currently many groups working on developing single molecule protein sequencing techniques, trying approaches like fluorosequencing, nanopore sequencing, and tunneling spectroscopy. But so far, none of these approaches can hold a candle to mass spec when it comes to reliably differentiating peptides and amino acids. So why not just use mass spec to sequence single protein molecules? So here's our dream of how single molecule protein sequencing could work. A denatured protein would be pulled through a nanopore ion source while it is fragmented into its constituent amino acids. The amino acids would then be ionized, focused by a set of ion optics, separated as they pass through a magnetic sector, and then detected by an array of single ion detectors. The sequence could then be straightforwardly reconstructed by considering the time of each detection. Now, I'm sure you're thinking that this seems like a far off dream, but I hope to convince you that it's not quite as far off as you might think. Not sure where I need to point. Yeah, I'll just take that mouse. Thank you. So, to bring this back down to earth for a moment, here's a standard proteomics workflow that I'm sure you're all familiar with. Now for us, the key to pushing towards this goal of single molecule protein sequencing by mass spec is all about this ionization step. So that's where I'm going to focus on. So I'm sure most of you are familiar with electrospray ionization, or ESI. Now ESI is a wonderful soft ionization technique without which most modern proteomics workflows would be impossible, but it does have a major limitation, which is its poor ion transmission efficiency. Now just to give a very brief overview of how ESI works, you have a high voltage, which is applied to a capillary, which contains your analyte solution. This high voltage causes the meniscus to deform into a structure known as a cone jet. The tip of that cone jet emits a spray of charged droplets. These charged droplets then undergo a drying cycle where they collide with background gas particles, causing them to evaporate and shrink into smaller and smaller droplets until eventually you are left with just bare ions. Those ions then need to be transferred from the high pressure region on the left to the low pressure region of the mass spectrometer on the right through a transfer capillary. And this is the stage where the vast majority of ions are lost. And this generally results in ion transmission efficiencies well under 1%. Now, as we've heard over the past 2 days, researchers in the single cell proteomics community are still able to achieve amazing results with such low ion transfer efficiencies. But to sequence a single molecule, we would need to detect every one of its components. So only detecting less than 1% of the ions we produce is totally unacceptable, and we need to find a way to fix this. So let's take a closer look now at the ionization process in ESI to see how we're going to tackle this problem. Now, this is a famous image from a paper by Kabarle and Tang, which shows the life cycle of charged droplets in ESI. Now, the charged droplets evaporate and shrink until the electric repulsion of their charges overcomes surface tension, producing a jet of even smaller, more highly charged droplets. This is shown by this inset here. So electrospray really has this fractal-like character where smaller and smaller charged droplets are emitted from parent droplets until eventually you reach the smallest nanometer scale droplets shown down here. And here's something very interesting happens. Instead of producing yet another smaller droplet, the charges are packed so tightly together and the electric fields are so high that ions are able to just pop right out of the droplets and into the gas phase by a process called ion evaporation. Now here's the main idea. If these smallest droplets can emit ions directly, why don't we just skip this whole messy and lossy droplet drying process and jump right to the ion evaporation? So what we've done is create an ion source that exists on the same scale as those smallest droplets that I've just mentioned. These are just pulled quartz capillaries whose tips essentially constitute a nanopore with an inner diameter of about 30 nanometers. And here you can see an SEM micrograph of one of our ion sources. Now, using these nanopore ion sources, we can achieve ion evaporation directly into high vacuum from the meniscus at the tip of the ion source, getting rid of the need to use a background gas and evading the primary modes of ion transmission loss in ESI. Now, in all the experiments I'm about to show, we use this homemade quadrupole mass spec on the right. And to be clear, this is not the magnetic sector sequencing instrument I showed in the first slide. This is just the mass spectrometer we are using to test these ion sources. But what I want to emphasize about this instrument is that the ion source is housed inside the vacuum chamber, right along with the quadrupole and the detector. So again, there's no background gas. So the conventional electrospray process where charged droplets are dried out to produce ions would be impossible. So as we were experimenting with ion sources of different sizes, we noticed an interesting trend. The smaller we make our ion sources, the nicer our spectra become. Here I'm showing mass spectra of a 100 millimolar solution of arginine obtained from 3 different ion sources with different pore diameters ranging from 300 nanometers down to 20 nanometers. And we can see that for the larger pores, we get a series of peaks starting here at, whoops. Oh, can I go backwards? There we go. It's good now. So, okay, so for the largest pore, we see a series of peaks, and the first peak corresponds to the singly charged unsolvated arginine ion, and each subsequent peak is separated by 18 AMU, or the mass of a single water molecule. So all these other peaks correspond to hydrated states of the arginine ion. When we get down to the smallest pore, there's basically just one peak corresponding to the singly charged unsolvated arginine ion. And we also see 2 much smaller peaks corresponding to the first and second hydrated state. Now, I should also note that when we tried ion sources with pores around a micron or larger, we are not able to obtain any spectra whatsoever. So it really appears that smaller is better. All right. So here we have the mass spectra for 16 different amino acids, all obtained from nanopore ion sources with diameters ranging from 20 to 60 nanometers. Each amino acid solution had its pH lowered below its isoelectric point by the addition of acetic acid to operate in a positive ion mode. And you can see that the unsolvated state of each amino acid ion dominates its spectrum, making the spectra extremely simple to interpret. And in a few cases, we can also see smaller peaks corresponding to the first or second hydrated states. So this ionization technique appears to be robust and work with the majority of amino acids, covering a wide range of chemical properties. Okay, so by now I've shown you a bunch of spectra of amino acids that we obtained using our nanopore ion sources in a homemade mass spectrometer. And my claim is that all of those spectra are the result of detecting ions that were emitted directly into high vacuum by ion evaporation from the meniscus at the tip of our ion source. But how do we know that that's really what's going on? How do we know that we aren't still producing charged droplets that are emitting these ions? So I will give you 2 arguments for this. First, it's well known that there is some minimum flow rate necessary to sustain a cone jet electrospray. And this is apparent just by continuity. You need to supply enough fluid to sustain the steady emission of droplets. But as you make your capillary smaller, the flow rate gets choked off by the constriction of the nanocapillary. And we find that for nanocapillaries used in these experiments, which are represented by the green arrow here, the expected flow rate is more than 3 orders of magnitude smaller than the minimum flow rate necessary to sustain a cone jet. So if we don't have a cone jet, we should not be emitting droplets. Okay, so that's all well and good, But what if droplets are still being emitted by some other unknown process other than a standard cone jet electrospray? So even if that were happening, the droplets would almost immediately freeze in the vacuum and would not be able to yield the bare ions that we detect. Second, we can calculate the probability that an emitted ion cluster will collide with a gas molecule en route to the detector. We know the pressure inside of our vacuum chamber, which is about 10 to the minus 7 torr. And to be conservative, we assume the worst case scenario of how many water molecules are evaporating off of the meniscus and might collide with emitted ions. We find that about 98% of emitted ions would experience no collisions whatsoever and travel along ballistic trajectories from the ion source to the detector. This means that there would be virtually no collisions to provide the kinetic energy necessary to desolvate a droplet or ion cluster. And so amino acid ions were likely emitted from the source in the exact same solvation state as they were detected in. So coming back to our vision for single molecule protein sequencing, the ability to emit amino acid ions directly into high vacuum is what puts this dream within reach. The near absence of any collisions means that the spatial ordering of emitted ions would be maintained after emission, allowing the sequence to be reconstructed. Now, there are obviously some technical hurdles that remain in order to realize this vision, namely in being able to fragment a protein into its constituent amino acids at the tip of a nanocapillary. But the ability to ionize amino acids and small peptides from aqueous solutions directly into high vacuum constitutes a major step towards this goal. So one last note, since I'm sure many of you are curious, we have tried this ionization mechanism with some small peptides, namely glutathione and two of its post-translationally modified variants, S-acetylglutathione and S-nitrosoglutathione. And we are able to successfully collect spectra of these as well. So here are the overlaid mass spectra of the 3 peptides. And again, we see that for each one, the unsolvated singly charged state dominates the spectrum, and we get some smaller peaks corresponding to first and second solvation states as well. So to conclude, we've developed a nanopore ion source that is able to emit amino acid and small peptide ions directly into high vacuum. This bypasses the primary modes of ion loss inherent to conventional electrospray ionization, which paves the way for single molecule sequencing by MS and could enable a significant increase in the sensitivity of MS-based proteomics experiments. So thank you all for your attention, and a special thanks to Oxford Nanopore Technologies for funding this work, and I'm happy to take any questions.
Thank you, Nicholas. So we have a question from the virtual audience. Excellent work. How many runs can you do with one tip? I'm concerned about the possibility of clogs. Have you evaluated the durability of this source?
Yes, so clogs do occur. It seems there is some variability between tips. We have used, so for example, if I can go back here. So all of this data was obtained with just 4 tips. So we have a system where we can sort of cycle through solutions while the tip is inside the vacuum chamber. And so when a tip is working, it tends to work for quite a while. And we can run one solution, then flow in a different solution, flow in a different solution, and continue for several hours until eventually the tip clogs.
So when you say flow a solution inside of the vacuum, you don't want to have water molecules, or do you actually have water molecules that immediately evaporate inside of the vacuum chamber?
So the only place that any water is exposed to the vacuum is at the tip of the ion source. So it's
then it immediately evaporates?
Well, there's water continuously evaporating, but it's not sufficient to cause freezing, or it's just such a small surface area that it doesn't really constitute a problem. But that is the reason we think that bigger tips don't work. So when we try tips that are a micron or larger, they tend to not work. And we believe that's because it's freezing because of evaporation.
John Yates has a question. How does the tip diameter compare to the old nanospray tips? How do you pull them?
So the nanospray tips, so the nano refers to the flow rate rather than the size. So nanospray tips, I think we're about between 1 and 10 microns. And so these are at least an order of magnitude smaller. We pull them using standard Sutter instruments, capillary pullers. It was just a matter of finding the right recipe, which I'd be happy to share if anyone would like to email me.
So yeah, I can see a lot of questions coming from the in-person audience and the virtual. I'm going to ask one more from the virtual and then go with in-person audience. So do you see to couple a separation system to this type of delivery of very flow rates required for nanopore tips? Would it be possible to combine it with liquid chromatography or capillary electrophoresis?
That's a great question. We have not tried that yet, but it is certainly something we would be interested in doing eventually. To be honest, I haven't really given too much thought to what complications would arise when we try that. But I can imagine that the very low flow rates could end up being an issue.
Yeah, it's certainly hard to imagine protein analysis being practical without some good form of separation.
That was awesome. I have a question that relates directly to the slide you have here.
Yeah.
What happened to tryptophan?
Yeah. So these peaks correspond to just clusters of water. So like a hydronium ion with a few other waters attached. These sometimes appear. Tryptophan is the only one here that was done at a lower concentration than the others because it's less soluble. So that may have played a role. But we don't fully understand why these water peaks appear in some spectra and not in others.
Gotcha. And I realize this is early, but
Yeah.
relating to the last question about clogging, proteins, as you know, have various solubilities and you can unfold them with various things, but they do have a tendency to stick to things. And I'm just wondering, do you guys have thoughts about how to make a system that's this small durable in the long run? Because I mean, you went through lots of capillaries in a concentrated solution.
There's definitely still some work to be done there. I'm not sure I have a great answer for you. There has been some work done with slightly larger capillaries. I forget the authors of this paper, but I'd be happy to share it. That found that actually, like with small capillaries on the order of hundreds of microns, proteins tend to actually unfold near the tips, which is a positive sign. But yeah, there certainly could be some issues there.
So that's a brilliant idea. If you can make the ionization efficiency from 1% higher, it would be a dramatic improvement. But one of the main problems with an ion source is, or one of the main functions of an ion source is generally to get rid of the neutrals. And so a lot of times you've got neutrals that are not ionized and they cause ion suppression. And so you lose a lot of your ions by the either, we can just put them in the category of matrix components and things that are just not ionized, and they'll knock off a lot of the ions. So how do you deal with, I assume these are very clean amino acids that you dealt with, but any sample that comes from a biological experiment typically has a lot of junk or matrix components that are not ionized. So how would they not cause problems once they get inside the mass spec?
So I may need to think about this a little bit more, but it's my belief that with this ionization mechanism, I don't think those neutrals would come out in the first place, because I think those come from droplets that dry out. If something's not charged here, it's not going to be pulled out of the ion source. I hope that answers your question, but I'd be happy to talk more about that.
I was just going to say, you'll find out if you do an actual biological experiment and run it through you.
All right.
Yeah, well, I'm afraid they will clog, first of all, the way they get into with any real samples. But a couple of questions. I have about 20 minutes of questions, but at least two main ones. How you push this thing in? Because you said you push in, but there is no chromatography, nothing. How you actually push them in, and what this was colored yellow thing in your picture, that how you separate amino acids. If you go back to the all the way down to the picture where you kind of show how you single amino acid just detect on the here. What that yellow thing?
Yeah, so that's supposed to represent, I guess, basically the fragmentation. So
how exactly is it? Laser short, or you just?
So the idea is to use a UV laser, and we're starting to investigate the possibility of doing that. That's sort of an ongoing project right now. But yeah, the basic idea is to use something around 193 nanometer light which has shown some pretty high efficiency at breaking peptide bonds.
And then you assume that the amino acids will just fly into it and then detect it on the detector. The second question, you said in the source you keep 10 to the minus 7 torr. It must be pretty big, turbo pump there.
Yeah, we do have a turbo pump. I'm not sure I understand the question. Sorry.
Well, how you maintain this? You spray something from atmosphere to 10 to the minus 7 and don't have any kind of problems.
I think the reason this works is because the tips are just so small. The amount of water evaporating off the tip is just minute.
Okay.
Thank you, Nicholas.
Thank you.
That's certainly exciting. I think there certainly have been claims of electrospray being much more efficient than 1%. Certainly Dick Smith would like to say they can make it 50% efficient. And people have also used very small tips with chromatography, which improves ionization, but makes the process more painful. So I think what you're showing is really exciting, as you can tell from the questions. I'm sure there are more questions in the virtual audience. They just want to get to dinner.
Yeah, sure.
Otherwise, we would stay here longer. But I think if you can make this practical, it would be a very exciting addition.
Sure.
Thank you.
Thank
you
you
so much for your presentation.